Welcome, Dr. Saurav Mallik. Dr. Saurav Mallik from the Department of Pharmacology and Toxicology, the University of Arizona, United States of America. Uh, Dr. Saurav Mallik, we welcome you from the portals of Hooghly Engineering and Technology College, as well as from IQAC, IIC, and EDC of Hooghly Engineering and Technology College. We are really privileged to have you here, despite the time difference from the other side of the globe. You have really been kind enough to join this faculty development program. Initial session, we once again cordially welcome you. Thank you, Dr. Saurav, for being here. Let me formally introduce Dr. Saurav Mallik. He is currently a research scientist in the Department of Pharmacology and Toxicology at the University of Arizona, United States of America. His previous experience includes serving as a postdoctoral fellow in the Department of Environmental Health at the Harvard T.H. Chan School of Public Health in Boston, United States of America, during the time period 2019 to 2022. Prior to that, he was associated with the Center of Precision Health, School of Biomedical Informatics at the University of Texas Health Science Center in Houston between 2018 and 19, and the Division of Biostatistics, Department of Public Health Sciences at the University of Miami Millard School of Medicine in Miami, Florida, USA between 2017 and 18. Dr. Mallik earned his PhD in Computer Science and Engineering from Jadavpur University, Kolkata in 2017, with his research conducted at the Machine Intelligence Unit, better known as MIU, of the Indian Statistical Institute, ISI. His tenure at ISI also included roles as a JRF in the Department of Science and Technology, Government of India sponsored Sharno Jayanti project, and as a visiting scientist between 2011 and 2013, as well as between 2015-16. Additionally, he worked as a UGC Research Fellow in the Department of Computer Science and Engineering at Jadavpur University between 2014 and 17. Dr. Mallik has been honored with a research associateship from the Council of Scientific and Industrial Research, CSIR, Ministry of Human Resource Development in 2017. Saurabh Mallik, he has got a vast experience and has worked in different disciplines in different corners of the world. We are really privileged to have him here in this 1 week faculty development program. Welcome, Dr. Saurabh Mallik. So the first session begins here. I would request Dr. Mallik to start the deliberation. Over to you, Dr. Saurabh Mallik.
Thank you so much. Good morning, everybody. I think my voice is audible, right?
Yeah, it's audible, absolutely audible.
Okay, great. So let's start. Uh, can you see the screen?
Yeah, the screen is visible now.
Great. So today my topic is uh, it is actually the interdisciplinary subject. Uh, is the understanding bioinformatics and computational biology through machine learning and deep learning and somewhere from optimization. So I'm basically not going to the details here because it's a vast subject. So, but the thing is that for the layman, we have to understand what is the piece, uh, area where this machine learning and deep learning, also the data mining, can be used. Because the thing is that now the research has been divided into two parts. One is about the medical problem solutions, and another is the security and networking. This is the main major two hubs, and we are in between that. If two things are basically two, which can be used in that. We are either in the we are all mathematics. These two medical domain to solve their problems, as well as if you want, you can use EO, that means application, different application computational approaches in networking, IoT. This is the lot of topics. So basically, I am discussing all these computational approach and how to do the bioinformatic analysis, and that should be the meaningful to the doctors and biomedical researchers. It is not like that we can do just 1 + 1 equal to 2. It is not like that, right? It should be meaningful to the biologist, and that is our challenge. So today's site map is first. Hello, is it audible? Hello, is it audible to everybody?
Yes, sir. Yes, sir.
Ye-
The slide is moving. Slide is moving.
Yes, sir, it is okay.
So now I am going to the second slide. Is it moving?
ও টেস্ট করছে একটু হেডফোনটা না
Okay. So today's in the site map today we will going to discuss about little part. So what is the site map or roadmap for this topic? First we will go looking to the introduction thing or basic the biologist. What is the biology term? Because everybody is not familiar with the biological term. So we have to know the basic terms, and also we have to understand what is the computational biology, and what is the machine learning techniques can be used, and then the future scope and conclusion. This is the things. So now I am going to the introductions.
Uh,
So
Excuse
in
me, Dr. Mallik, sorry to interrupt you.
yeah,
Sorry to interrupt you. If you could please make the screen a full one.
slide. That's why I'm saying the slide is moving or it is just stopped.
No, no, it's working, it's moving, it's absolutely, but we are not having a full screen. Many of the participants from their mobile phone. So if it is
So it is full screen now. I don't know what is happening in your. It is full screen now. Is it visible?
yeah, it's visible. Okay, you may carry on. You may carry on. It's not a full screen, but still okay. But it's working. You may carry on.
Okay. So the slide is moving, right?
Yeah.
Okay, great. So in the introduction, the thing is that as a machine learning persons, we have the basic things. What is the basic things? We all know about the feature selection, because today's era is the big data, right? There is a big data and multi-dimensional data, and it also happens to the biologist also. There is a lot of biology data. Just 2 years back, we just passed a critical moment, that is COVID. So we have a lot of data. It may be the COVID or cancer data or anything. So here the thing is that here the feature selection, here feature mean in the biological data, it should be used as a matrix. In the matrix, in one side, in one row, you can say it is the genes, and in the column, we can say as a the patients, right? So here features means the genes or any biological, that means objects that can be used as a feature. So the thing is that in the biology, the main problem is that the problem is the imbalanced data. That means number of rows or number of genes, or you can say the features, is very higher. Number of this there is very high. maybe the 1 lakh or 2 lakh or maybe 50,000. But in the patient number of patients or number of columns, it is limited. Maybe you can get 100 or 200, maybe the 500. So you can get imbalanced data. So the thing is that now our challenge is to reduce the number of features. That means reduce number of columns. Rows, to make it. That means the doctors very that means useful as well as less costly, because we can't use 1 lakh features or genes to determine the disease. So if we can do, we can, that means provide 50 or 100 significant set of features or set of genes by which that features set we can easily predict the genes or maybe the classic. This is the classification. So thing is that first is the feature selection, and another is that feature extraction. It is a different things. Feature extraction is a different things. That means we can create a new features using the existing features. That is also big challenge. Second is the classification. This is classification, right? Suppose you go to a that means hospital. Somebody has a that means cancer. Somebody does not has a cancer, or maybe the cancer. Suppose in the cancer there is a lot of tissues. If you go to the that means brain cancer, this is a different tissue, and there are different subtypes. So there is a different challenges. Okay, so this belongs to this classification. This is classification stage, and the last one is the clustering. There is a some that is latest research which is called the single cell sequencing. In that data, this is also biological data. There, the thing is that we don't have the labels. Like we don't know which that means patient or class belongs to cancer or non-cancer, because we work on the cell rather than the patients. So in that case, this clustering is used. That means where we don't know the clusters. Okay. So this is the major things. Now I'm going to the biological concept. Now we have to understand what is the central dogma of molecular biology. There is the three basic term we all know, that is DNA, deoxyribonucleic acid. Second is ribonucleic acid, and third one is protein. We all take proteins, like that means all the foods are in the protein level, and DNA is the most stable level. This is the called deoxyribonucleic acid. It is a double strand, and when it that means breaks into single strand, it is called the RNA. Okay, and it is unstable, no, not more stable. And we know the vaccination, we take the vaccination, and that vaccination belongs to this RNA level. That is, it is called the RNA vaccines. That means we may think that using the vaccination, that means disease cannot be totally that means forfeited. It can be reduced, but because it is unstable, you can understand this is the RNA level vaccines, right? So this is the things, and there is a three terms are there. When this DNA to RNA conversion will be there, it is called the transcription. Another is the translation. When RNA to protein, right? And another is the replication, DNA to DNA. This is the basic things. I'm not going to the details. Another thing is very important things. We already discussed about the features. What is the features here in the biological data? The genes or microRNA, any biological molecule belongs to these features. And another important, that means biomolecule, that is called the link RNA. Link RNA is the non-coding RNA. Normally, gene is actually part of the that means some area of the messenger RNA, that is called the mRNA. That means it carries the that means some data, some informations, right? But here it is called, there is a two term, that is actually not the coding RNA. That does not that means they are not carrying any data or nothing, but they has the influence to on the genes to make the diseases. That is the things. So they are just like an enzyme, just like the influencer, which has a tendency to reduce or influence the genes or messenger RNA. These two are long. One is link RNA, another is microRNA. MicroRNA is small non-coding RNA. The thing is that microRNA actually targets a lot of genes, okay, which try to reduce the expression level of the genes. Just like a heartbeat, we have the heartbeat, we have the heart, we have the heartbeat. That means it going up and down, not in the normal stage. Similarly, in case of that means cancer or any disease, these genes levels is simultaneously going up and down, and this makes this dysregulate. This is called the dysregulated genes, and this makes different disorders or cancers. This is the disease. So our main target is to find out those features which has a tendency to make the cancer, and these two that means influencer has a that means separately has a influence on these genes. Okay, so it is a multi-dimensional, multi-dependent biomolecules. Okay, so here you can see the H19 is that means one type of link RNA, which is that means has a big role in cancer detections or any others. So today I will discuss 2 or 3 works that by which you can get understand how we can use this that means our that means computational approaches on this biological data. So now I'm going to the what is the computational biology and bioinformatics. I already said that it is the interdisciplinary subject. So basically things the biology and some computational things. Computational things means it should be some novelty on the methods, or you can make a measures, or you can that means add something in the measure. Okay, or you can that means make a framework using the existing that means steps. Okay, and these steps may be some portion will be that means belongs to computer science, some will be the deep learning, some will be the machine learning, some will be the data structure also. Okay, you know the hashing, that is a that means this is a very commonly used from the data structure, and also some hypothesis test, that is called the biostatistical test, also used here. This is also belongs to this computational biology. Okay, and this all these subjects, that means computer science applications, methods, biostatistics, and so data structure, all this can be used to make a that means make a big role to analyze the data, and it may be the combined effect, and that is the big thing. Okay. Now I am going to some portion of the feature selection, how it goes. So feature selection is a process, we know, to select a set of features that can effectively distinguish between the different classes or clusters. Okay. And the thing is that what I said, that most of the features,
Hello. Hello, Dr.
yeah,
Mallik.
yeah,
Very sorry for the interruption. Actually, the slides are not moving. Can you share the screen, entire screen, in entire window? Actually, I think now it
I
is
actually
moving.
see. Now
Uh,
is it moving?
it is moving, but while sharing the screen, please share the entire screen.
When I that is the problem. When I make the inter-screen sharing,
Hmm.
the slide is not moving. Okay,
Okay, now
I
it.
don't know.
Okay,
Now
okay,
it is
now.
moving, right?
Yeah, yeah. Now the slides are moving.
I'm
Yeah.
just trying to zoom by which
Okay,
everybody
okay,
can.
okay. Okay,
I
okay.
don't
Now,
know
now.
what is happening. Yeah,
Yeah, yeah. Now it's fine. Now it's fine.
great, good.
Please continue.
Yeah. So in the feature selection, what I said, that there is in the data, if you see the biological data, it belongs, it just like look like a matrix. Here, what I said, that in the row, you can say as a features or genes or microRNA, anything biomolecules, and the columns can be says as a patients or cells or anything. So the big challenge is that we have to reduce the number of rows, because there is a lot of maximum that means features are irrelevant, and it increases the time complexity and other things. They are the most redundant features. That is the things. So we have to remove this. So to do this, there is a basic things we have to understand. What first thing is entropy. We know all entropy. What is that? Entropy means if we toss a coin, then that means what is the uncertainty ratio of the percentage of uncertainty, that is half. That is a you can toss is the head or tail. If you say the how much uncertainty is there to make the head, it is 1/2. There is a two occurrence and one is happening. So this uncertainty is called the entropy, and entropy has a major roles to compute the feature selections here. Okay, we all know that means Shannon entropy is there. We know that This is the that is formula. I'm not going to the formula. Now I'm going to another topic that is called the mutual information. So mutual information means actually mutual information between two random variables. Here variable means features, feature vector, or you can say to the arrays. If you have the two arrays or two vectors or feature vectors, we have to find out the mutual information. So mutual information means which is common. That means which is common and which is that means common things. Right? Here the thing is that if you have x and y two that means vectors, then the mutual information we can say denoted as a ixy, and here there are three features are there. One is it should be greater than equal to 0, that is non-negativity. Another is that if x and y are totally different, they need to be 0. That means mutually exclusive, then it will be 0. And it should be the same. If it is the symmetric, then it will be 1. That means these are things. Now I'm going to the supervised feature selection techniques. So very well-known feature selection technique is called the MR. I think everybody who are working in the machine learning, I think they should be aware of this MR technique. So here the two things are there we have to understand. One is that if you see the formula, there is a two part. This is I, Fi and C. C means the class variable, right? Class variable means class vectors. Suppose we have the 100. Suppose we have the 100 columns. That means column is if you have the 100 that means patients. Suppose among the 100 patients, suppose you have 70 cancerous patients and 30 are non-cancerous. That means normal patients. So the C should be, if you that means denote all the cancer patients as 1 and non-cancerous as a 0, then it should be the combination of 1 and 0, which has a size 100. Okay, so this is that means the class variable. And here the Fi is the, there is a two bucket. It should be the two bucket initially. That means the you can see the S for that is final. You can say the final, and I is the initials. Okay, so initially all the features should be in the initial bucket. That means in the destination bucket, that is called the S here. I don't know the that means here actually destination bucket. Okay, that is selected buckets, and that should be vacant. Here the idea is that we have to select each features from the initial bucket, and we have to that means find out the mutual information between the initial bucket and the class variables. Okay, that is called the that is relevance. If the relevance is higher, hello.
It's okay. You may carry on.
Yeah, okay. If the relevance is higher, that means that feature has a higher prone to that means predict the class variables properly. Okay, and second part in the right hand side, you can see the another part is I, that belongs to which can find out the mutual information between the each features from the initial set and the selected feature, each selected features from the final sets, that means selected sets. Initially, when we pick up the first features from the initial set, then right side should be 0 because Fs in the Fs there is no features, right? So it should be 0. In the left side, it should be the that means value between the class variables. That means the mutual information between the class variables and the first features, that means random features. And here this is the optimization. This is called the optimizations. Okay, and we have to run for the all features. Okay, and here you can see the S. S is the size of the set of the already selected features. Okay, that's the things. And here in the MNMR, you can see it is the single objective function where the redundancy should be omitted from the relevance. Okay, and the thing is that here you can see That means this fraction of redundancy, that means can be changeable according to the different weight factors. Right? Here, they, as a weight factor, they are taking all the size of the already selected has a weight factor. But when we going to the next slide or next methods, you can realize that you can change it according to your requirement. In the basics, in the when this feature selection, there is a basic feature selection technique. When it is first discovered, they are using this. This is the optimization technique, single optimization technique. Okay. Now another similar that means feature selection technique is there. It is called the input normalized mutual information is feature selection. Here you can see they are using R. R is, that means they are using the different that means uh, different things or different criteria to make the weight as a different, but the other things are same. Okay. Another method is DFS. It is also similarly they are optimizing using the single objectives and just changing the weights. Okay, and another is there is a lot of things. I'm not going to the details. And another methods is it is called the BWMRMR, where similarly they are using some that means weights. Here you can see some weights are there. Similarly, it is the recent 2015. So I think you can understand how can we that means compute the feature selections, and it should be between the two groups. Okay. Now the what is the evaluation criteria for this that means features? Suppose we find out the features. So what is the evaluation criteria? There is the three criteria. Basically, we can use the classification accuracy, redundancy rate, and the representation entropy. But there are a lot of you can use whatever whenever you need that. It is the I'm not going to the details. Okay, these are just that is just criteria to how to find out this that is measure of the features and some related feature selection method. If you want to use first method, you can see it is the published in the Cybernetics. IEEE Cybernetics. It is a very good journal, and you can see first papers is the identification of multi-view gene modules. So modules means the clusters. That means previously I already discussed there is a three things are there: features, set of features finding or feature extraction or feature selection. Second is classification. Third one is clustering. These first papers, I'm not going through the details. This first paper basically find out the that means features. That means features first, and then to find out the gene modules. Okay, that means set of clusters of genes or cluster of features using this that is mutual information and some graph theory. Okay, and the second paper you can see it is published in the IEEE Nanobioscience, where what is happening here, we are finding the epigenetic biomarker. So epigenetic, what is that? I am saying just a little bit what is epigenetics. So thing is that biomarker means gene marker, that means the gene features. So why it is epigenetic? Epigenetic means like gene expression data, there is another thing is that it is called the DNA methylation. So it is the part of the epigenetic. Epigenetics means where the gene sequence does not change. That means genetic information does not change, but according to the environmental effect, some added situation will be there. Like we are now last 8 years I am in USA. So in the genetic that code of my body is not changing, but due to the environmental effect, obviously my body is changing. Okay, that is called epigenetics. And why epigenetics is there? Actually, the changes it is happening after 5 or 6, a minimum 5 years. So if we try to find this epigenetic biomarker or epigenetic genes, maybe using the DNA methylation data, that would be more stable marker or genetic features or whatever you can say. And here we are using this maximum relevance. What I said, that relevance is should be maximized and the redundancy should minimize. Why redundancy should be minimized? If both the features has a similar kind of dependency, that means we can take one features. Why both two? This is the idea of the biological perspective. That's why if the redundancy is higher between two features, we try to that means minimize that. Okay, and relevance is if the feature single feature has a higher prone to classify that class levels, we can take that features more. That's why we are taking the maximum relevance and redundancy should be minimized together using the feature sequencer for multiomics. Multiomics means what is omics? We know that there is a genomics, proteomics. Okay, there is a all the data which ends with omics, that is called the omics data. It may be sometimes if you have suppose you go to the hospital generates the gene expression data. Suppose suppose gene expression data maybe for maybe the brain cancer, right? And also simultaneously for the same set of patients, they are also providing the environmental changes data, that is called the DNA methylation data. So we have the both the data. There is a lot of other data are there, CNV, copy number, variation. We have all the data, but the common thing is that maybe we can get some common features, common genes in the both the cases, gene expression and methylation data. And also the patients, we can get maximum common patients should be there, right? So we have both the things together. Okay. And using this multiomics data, we are trying to find out the stable biomarker that is called the epigenetic biomarker. And the third one is that, it is called the integrated strategy of hierarchical clustering for the singular value decompositions. It is also biomarker discovery method for the acute myeloid leukemia. But the thing is that here we are using, that is some framework using the SVD and the higher clustering, and that is published also. This is also all our computational approaches. That's why I'm describing here. It is published in the IEEE TCB. It is a very good journal. And another paper, that means it is paper which has been published 3-4 years ago, that is using this H19, that means long coding RNA, okay, using the transcription factors and also the genes. That means here the thing is that there is a multiple molecules is there. Our target is to find out the gene set of genes or set of features where the other dependent factors are there. What is the other dependent factors? Here the microRNA try to reduce the gene expression level. That means inverse relationship between the expression level of the microRNA and expression level of the genes. So what one impact, that means influencer is the microRNA. Another influencer is the transcription factor. Transcription factor is basically a protein which can reduce or which can amplify. It can both the things. We are not sure. According to the conditions, it can influence or it can suppress the gene expression. Okay. Another thing is that that is LcRNA, long non-coding RNA. That also has a influence to that means make higher the gene expressions. Okay, sometimes it's also reduced. So here the multiple influence on the gene expressions. Okay, and here we are trying to find out some genetic marker as well as the feature models. Okay, so it is published in the nucleic acid research, that is journal. It is also good journals. So what is the, I'm just describing the this method because it is very good bioinformatic analysis will be there. So what is the background and what is the method for these motif finding papers? What is that? The background is that what I said, that link RNA has recently been found to be most important in gene regulations. Link RNA H19 is one of the link RNA that has been reported to play a oncogenic role. Oncogenic role, that means which can cause cancer. That is called the oncogenes. That genes which causes cancer is called the oncogenes. So H19 has a big role to make the oncogenic roles in different tissue-specific human cancer. Its specific regulatory role is still exclusive. So here, what we are trying to do in this study, we developed a novel analytic approach by integrating the synergistic regulation among the link RNA. Here we are taking the H19 transcription factors, target genes, and microRNAs, and then applied to the different pan cancer expression datasets from the TCGA. TCGA is uh, online available, very popular datasets where we can get this omics data, that means biological array-based data. It is called the array-based. Actually, biological data is two type of data. One is imaging data, biological imaging. We know that brain imaging, brain MRI is there. This is one kind of data, biological data. And here, what we are describing here, this is called the array-based data, where some values are there. There is no images. Okay. So TCGA provide this array-based data, that means matrix-based data. Okay. And second thing is this identification of a triplet that has a that means this is called the gene regulatory network, where the genes is there, TF is there, and H19 is there, and that is also mediated by the microRNAs. That means 4 factors are there. This is the that means total diagram. So we try to first win the TCGA data, where we are try to find out 24 types of pan cancer data. Okay, and from that data set we are trying to find out the link RNA expression data, mRNA expression data, and then there is a transfact and trust. Two data set is there which can provide the that means mapping between microRNA and genes. That means which microRNA target which gene, that provide that things we can get from the different target minor data sets. And also by this transfact and trust we can get that means which TF, which protein can that means target or maybe the influence of which genes pair, that can get from this transfer and trust data set. So we have now link RNA expression data set, microRNA data set, and then we are trying to find out the TFs and gene pairs. That means which genes is that means influenced by which TFs. TFs is, it may be protein or not. Sometimes it is protein. Okay. Now the thing is that there is a two part which we are trying to the focus on the H19 expression levels. Now we are taking the lowly expressed samples, H19 samples, and highly expressed samples. Both the cases we are trying to find out the which is the H19 corresponding, what is the TFs and genes. We trying to get, this is the histogram. You can see in the right side the red color is higher and prominent for the H19. That means it is the highly expressed, and the in the left side you can see the green color. It is called the heat map. Okay, red means highly expressed, and the green means lowly expressed. And in the cancer detection or any disease predictions, this highly expression and lowly expression make a big role. If it is the that means normal, it is the not big red, not good. That means big green. Then that is a C. That means the expression level is almost that means normal. So normal things does not make sense. Okay, if it is the abnormal, it may be higher and lower, then it makes sense. It can create some disorders. Okay, and then we are using the linear regression technique. Okay, and we try to find out some that means models and try to find out some coefficients. So using these coefficients, we can predict the other data. That's the things. So here we are trying to find out the models, as well as we are trying to find out the regulation triplets. Okay, there is a two cases has happened here. First case is the when H19 is lowly expressed for which samples we are taking those. Okay, and also similarly where the microRNAs, MIR mean microRNAs. Okay, microRNAs where the microRNAs is highly expressed. Okay, and also the TF or genes should be lowly expressed. As I said, the microRNA try to reduce the gene expression levels. Okay, it should be the inverse. Similarly, H19 has a normally it can be as well influencer. That's why I'm saying that. So, which as a opposite direction of the expression levels, we are taking that. For one case, we are taking first case we are taking the H19 lowly expressed, and similarly the other things also. And in the second case, we are taking the H19 highly expressed, and we are taking the three combination. This is called the triplets. Okay, and then we are just try to do some experimental validations. Okay, and also the other things. I'm not going to the details. These are basic things. How can use the the computational things into the biological problem solving. Okay, so as I said, the data collection is collected from the TCGA. So TCGA, there is a 24 cancer types are there. This is a BLCA, BRCA, one is breast cancer. There is a lot of things. Okay, one is the liver cancer. There is a lot of tissues, and from each cases here we are taking 24. Okay, and just try to combine them. And also what I said that tissue specific data. Um, here that's a different kind of data set. One is FPKM data set. There is a different kinds of, you have to see this. Sometimes is the counter data, count value data. So that data is collected from the Xena browser. Okay, Xena browser is also that means combined with the ESC, SC, and also the TCG. It is already connected. And the third thing that we have to find out the interaction between the TF and genes from the transfer and trust, okay, which can provide the set of TFs connected with the genes. Another thing is that the interaction between the microRNA and the target gene should be found from the different target minor database. Here we are using the SpiderMeat. There is a tool is there which basically map between 6 or more target prediction database. So we are using this SpiderMeat, okay, which can that means provide the microRNAs, the pair of the microRNAs which has a which can target the genes. Okay. The last one, what I said, the H19 targeted microRNAs, that link mapping should be collected from the published literatures. So I'm not going to the details because there is a lot of works. I'm just describing some of the work and how it goes. And here you can see the that means there is a log fold change value and also the p value for the that means TF gene regulations as affected by the H19 expression level. Okay, and then also we can see another tables where which can define the TF genes regulatory regulation interactions significantly affected by the expression altered by the H19. Here you can see the cancer types are there. Is the number is there, TFs, and how many genes are there, what is the pairs, and what is the triplets. I'm not going to the details. It is the maybe the part of the biology. Here is the important things. What I said that upregulated. What is the upregulated genes? The genes which is significantly statistically significant. Okay, and which has a higher expression value on the cancer patient rather than the that is normal patient. That is called the upregulated genes. Upregulated genes and downregulated genes, that both are called the dysregulated genes. So both has a connections to make the disease. So here in the two groups, the first group is H19 lowly expressed set. Here there is a two modules. In each modules which are the that means biomolecules is there, and what is the p value. Things you have to understand. If the p value is 0.05, then we can say as a significant. So what is the p value? P value is the probability value by which that means any event can be happen randomly. That means here the errors, the percentage of the error can be determined by the p value. That means the p value is if it is the less value, then the chances of error will be less. That is the things. Obviously, there are other methods will be there by which we can that means correct the p value, but I'm not going to describe here. So p value is the p value is less than 0.05. We can say that biomolecule is called as a statistically significant, and these things we can do by different hypothesis tests like t test. There is a lima, ANOVA. There is a lot of tests. There is a different conditions according to the distribution. We can use that method accordingly and find out p values. And this is the regulations. How we can get with the regulations? By the fold change value. So fold change value is the mean value of the cancerous patient's expression value by divided by all the mean value of the normal patient's value. And here we are using the log to fold change. Okay. So this is the thing which biomolecules make the upregulation, downregulation. It provides here. Okay. So what is the result of the wet lab? You know that when we find out actually this work is a combinations of the dry lab and wet lab. So dry lab means the computational work. That means all the downstream analysis done by the computer scientists just like me. And also the wet lab. What is wet lab? When we suppose we make provided the results. Okay. That means this happens, this combination is happening, And when we get some set of features, that means we are saying that this 10 features, or maybe this that means combinations like H19sp1 has mediated by this microorganisms. Suppose we are giving 3-4 conditions, and we are saying that this combination or that we just giving result, that means this will happen, this will happen, but after we are that means giving the result, they will test in the lab, and sometimes it happen if it is the 50% chances of occurrences in the same things in the wet lab, then we can say that our result will be good. Okay, then there is a chance of publishing in the good journals. These two things is happening. So here the result, what is the result? This is the triplets. The H19 sp1. What is the sp1? Sp1 maybe the genes maybe, and one is tf maybe. So these are the combinations. Okay, I am not going to the other details here. Um, I'm not going to just skipping this one. So this is the things. This is the summary. How can we can use this? Okay, and another work. It will be very much interesting to you. This is that means one paper which is accepted. Maybe it is published in 2001. It is a very good conference in ICIBM. Here, that means we are comparing different supervised feature selection algorithm or method, okay, to find out the top features and gene signature from the multi-omics data, and it will be very much interesting. How we do? So this is the flowchart. So we have in the that means beginning, you can see we have the omics data. That means we have the gene expressions or methylation data. Maybe here we are using the gene expression data. So first, the thing is that when we try to that analyze any data, we have to remember in the data there may be some noise. Okay, so thing is that before using that, we have to remove the noises. So basically, what we do, we to try to find out normally as a pre-filtering stage. We try to find out we have to find out the variance for each gene vectors or feature vector, and we have to omit the genes or feature vectors which has a low variance, okay, or low standard deviation, whatever you want to say. And then the remaining that means highly variant genes, with the highly variant genes we have to proceed to the next step. And in the next step we have to find out, we have to do the zero mean normalizations should be, actually normalization is scaling, but that we have to remember that normalizations should be, that means feature wise, not totally. We sometimes it happens, we are suppose there is a zero mean normalization, mean max normalization, there is a lot of normalization will be there, but the thing is that we can't use the normalizations to the whole matrix. It will be wrong, but biologically wrong. So we have to use the row wise, that means normalization. That means if you have 30,000 initial features, that means rows, you have to use 30 times, sorry, 30,000 times the normalizations. Okay, so 30,000 times you have to use this, and then you have to use the Lima statistical test. What is that? Lima is the tool. Okay, and the thing is that Lima is the parametric test, one type of parametric test that that means that uses the empirical based test. Okay, and it try to find out the statistically significant genes and provide some p-values. Okay, and using these lemmas, the thing is that we can get some that means genes, statistically significant genes. Now we are trying to find out that means features using the traditional computational, that means computer science based feature selection method. Here we are taking the 5 methods: MRMR, TFS, INMIFS, VWMRMR, and SVM, RFE, CBA. Okay. So after that, what we do, we just using the 10-fold cross validations. Here I am using the 10-fold. If you want, you can use the Ligo now, or any 5-fold according to the, you can use, that means some that means measurement. to understand, that means to select which fold should be used here. I am using a 10 fold, and then we have used different classifiers. We have used the net base, KNN, Adaboost, and C45, right? And then we are using the different, that means performance evaluation matrices like accuracy and redundancy rate and representation in entropy, and for each cases. Okay, so we have to, that means here what is the basic things we all know, that in the biological data sets we use the different hypothesis test. And in the biological data, suppose if it is not the biological data, normal data, maybe the Facebook data or any social site data, we normally use the feature selection method. But here you can see there is a double filtering. That means you are using the statistical feature selection method as well as the traditional computer science based feature selection method together to find out the significant features. There is the good things. Also, in the biological perspective, we try to find out some intersection between top 50 significant genes, features, and Venn diagrams, and we try to find out some signatures. Signature means is similar to the biomarker by which we can classify the disease properly or any subtypes. Okay. So there is the four part. First is data collection, and then data, that means statistical test and find out the features. Then is classification, and four is, that means some, if you want to some do some biological experiment, okay, by wet lab or gene set enrichment analysis, that you can do. There is a four part. Okay. So I'm not going to the details. Here you can see there is a gene expression is there, exon expression is there, copy number and DNA methylation is there, and also pathway activity datasets. And here the three labels are there for the samples. One is favorable, one is normal, one is poor. Okay, profiles are there. This is very important. Okay, so if you want to use or analyze any biological data, multi-omic data, where there is a multiple or multi-view multi-dimensional data, you have to take the average accuracy. This is very important. Okay, so if you want to use or analyze any biological data, multi-omic data, where there is a multiple or multi-dimensional data, you have to pick out that is common samples. Otherwise, you can't work on that. Okay, this is that. You can see the with the pre-filtering, we have this data. These are basic things. I'm not going to the details. Okay. Now you can see this is result. You can see for the first is EXP means expression data. For five classifiers, we are getting, that means this accuracy, and the bold means, that means best accuracy according to the row. Okay, and in the bracket it is the standard deviation. Actually, when we run any classifier, the thing is that we have to run for 30 or 40 times. It is not that we have to run only 4 or 5 times. Okay, and here we are taking the average accuracy. This is important, and standard deviation is very important. If the standard deviation is very high, then it is bad. Okay, and if the standard deviation is very low, that is also bad. Okay, so standard deviation medium is good. So you can see the what is the result, and you can see the all the features when we use the emergent selection methods, feature selection method. For that feature selection method, using the C4.5 classifier, we are getting 71.32% average accuracy, where the standard deviation is 2.487. This is all the things. Okay. So this all the papers is combo of the computational things and the biological things. Okay, I'm not going to details here. Okay, another thing is that here you can add a comparative study between different feature selection method. Okay, here that means for expression data here, what we do, we are taking the VWMR as a, that means main method, and with the other method, other feature selection method, we make a comparison. Because the thing we try to, that means we actually the VWMR latest method, and it was that means developed from our group. So we have used this VWMR method to provide a summary of the results. Okay, which provide the better cases. You can see is the win draw loss cases, and that is VWMR versus other method. Okay, that means the first one you can see the 301, the first cell. It denotes, that means BWMR has a win 3 times. Okay, that means on this MRMR feature selection method, and one is loss and draw is null. Similarly, we have provided the summary result for the other cases, and you can see the maximum cases this BWMR provides the better result. So this is the things. This is the comparative study, and it is very important. Similarly, for other, this is the previous study. It is the average accuracy, and for the second one, it is the redundancy rate. There is the 3 criteria. So, 3 summary matrix will be there. Summary result will be there. Okay. Now I am skipping this. Also, this is the intersection of the 50 in extracted significant, statistically significant genes or features for the expression and exon expression data. Here you can see the 7 overlap gene signatures, okay, for gene expression data. Okay. That means here 1, 2, 3, or 5 sets are there. 5 sets means 5 feature selection. That means we are use different 5 feature selection method here. 5, not sorry, 6 or 5 maybe. And using this 6 or 5 feature selection method, we individually try to find out the 50 features, and we are trying to find out the overlap, whether which are common. If the common is there, that common has a major impact. That means error rate should be minimized here. You can see for the gene expression data, if we use the 6, 1, 2, 3, or 5 feature selection method here, and we can get the 7 common overlap gene signatures. So these 7 are very important. And for the XN expression, we can get the 9 overlap gene signatures. Okay, so this is the things. So I'm just skipping this. So what is the summary for these things? We use the BWMR algorithm that provide the best accuracy among the 4 datasets. Okay, that is biological datasets. Okay, and BWMR algorithm offers the best redundancy rate for the 3 datasets, while it provides best redundancy rates for the 2 datasets. Okay, so this is the summary, and this is the what I said in the previous slides, that is 7 gene signatures and 9 gene signatures is happening. Okay, now in the last stage, maybe in the last 5 minutes, I'm describe some of the things that has the good things. We know the what we discussed previously, that is called the RNA, that is called we all know about, maybe in the biologists, next generation sequencing data. It has a 2 type of data. One is RNA bulk sequencing data, another is single cell sequencing data. Okay, so what is single cell? Single cell means, instead of, suppose you are the patient, you go to the that means hospital, and you just sign the contract that your data can be used by the hospital. When you sign on that, your data obviously will be used by the for research. Here, if the single cell, what is happening, instead of patient's data, they are taking all the cells, not a single patient's. Suppose they are taking the cells from the heart, from the disease, breast, from the, that means, brain. For all tissues, they are collecting the cells. So here we have, that means in the column, instead of the patients, suppose patients, number of patients are limited, maybe the 30 or 100, but the number of cells will be a higher number, right? And the thing is, that challenge is that here the number of cells maybe goes to the 1 lakh, 5 lakh also there. So there, in that case, in the single sequence sequencing data, so we are taking each cells individually. But in the bulk, or previously used previously used next generation sequencing data, this is called the bulk sequencing data. What is happening there? You can see the difference. There we are taking average of all cells together. So our analysis might not right, but the single cells, what is happening? We are taking the expression value of each cells individually. We are not taking any average. So this is the big things, advantage of the single cell sequencing data. And the thing is that here there is a no problem with the imbalanced data set. In the bulk RNA, what is happening? The number of features are higher and number of patients are low. So that is the imbalanced data. So there is a lot of problems happens to that means in the single cell data, sorry, in the bulk RNA data due to this imbalances. But in the single cell data, there is no imbalances. But what is the problem is there? Here, class levels is missing. That means it is the unsupervised class, unsupervised problem. That means we don't have the class level. We have to find out the gene modules or clustering, cell clustering. Okay, that is the big challenge. Okay, I'm not just keeping this. This is the biological things. Okay. So this is one workflow. Okay, so first workflow is that we have collected the data and then we have to make the normalizations. In the normalization, we have to focus on the batch effect. What is the batch effect? Batch effect is that suppose when we collect the each cells, it may be happen from the 1 year all the cells are collected. So when every day is matters, so temperature is different. There's some bacterial effect will be there. So there are different factors. That is called the batch effect, and due to that some error will be there. So this is one thing, and also cell cycle effect is there. Okay, so the challenge is there. We have to make the normalizations properly, and we have to use the batch effect removal techniques, and also use the feature selection then, and then we have to use the dimension reduction that means technique. Sometimes we use the PCA, sometimes we use the TSNE. Okay, and then we have to use the clustering technique. Okay, actually feature selections is there. Feature selection means we can use the MRMR as well as some of suppose different t-test or lima to find out the genes. Okay, there is the things, but the thing is that before using this uh, hypothesis test, we don't have the class level. So we have to find out, we have to use the clustering first to know the class level, which cells belongs to which category. And when we get this category, then we can use this statistical test that is called the DE test. You can see the DE, differential expression test. Okay, so this is the things, and then after that we can validate. So in the single cell sequencing data, the added thing is there. What is the challenge? And another thing is that in the single cell data, what is the big problem? There is a lot of zeros are there. We know the sparsity. Sparsity means maximum values are zero. The single cell sequencing data has the problem that it has lot of zeros, and we don't know which zero is real and which zero is a dropout or make the by mistake. It is happened by mistake, and we have different imputed matrix strategy by which we can correct this that is dropout zeros. Okay, that is the things. Yeah, what I described here, the sparsity is the major problem, and to find out the dropout, find it. Dropout means which zero has real zero, which zero is a made by mistake. Second thing is that optimal cell cluster identification. Clustering is important to get the that means uh, class levels, and when we get the group level of class levels, we have to use different differential expression method to find out the biomarkers. And then obviously we can use some that is reduction of the time complexity method by using some searching method like hashing or other, and then the other regression method. That thing is there. Okay, so this is the things, the technique. First we collect the data, and then if the data transformation is needed whenever it needed, and then we have to make first that means cell clustering, and then the feature selection after that, and then we have to find out the markers, gene markers. And when we provide the gene markers, the thing is that suppose in the cell clustering, suppose we get multiple cell clusters, suppose we get the 5 clusters. So here the 5 groups are there. So how can we use the t-test? Suppose we are using the t-test. The t-test normally uses 2 levels. That means it provides the p-value. It compares the between 2 groups, not the multiple groups. So what we do here, we using watch one group versus rest group. We have to that means repeat it for the 5 times if you have the 5 groups, and then we have to find out the common that is significant genes. These are things. So it is very useful and hot topic, because thing is that the sparsity is a big problem, and we can use, there is a lot of hashing technique is used to find out the droppings. Sorry, dropout. It is called the imputed matrix. Okay, so hashing is very, LSH is very important here. Sometimes use it with the LSH. Okay, So the things that not just skipping this. Okay, this is the all the other methods. Okay, this is the graph based clustering. If you want to do this, okay. So it is all the measures. Okay, I'm just keeping this because time is very limited. So that is the called locality sensitivity hashing. It is a very useful for single cell data analysis for dropout finding and cell clustering. You can see the dropout, how it is happened. We have to find out which zero is original, which zero is by mistake, and there are a lot of methods are there to find out the dropout finding, that is called TRI impute, SCL impute, KNN impute. There is a lot of methods. I'm just mentioning here. What is the conclusion and future work? Multiomics single cell clustering. We already described previously, I have described how to analyze the multiomics data. That is called the bulk RNA, not for the single cell. In the second app, we describe only single cell sequencing data. But if we have gene expression, methylation, or other multiple, multi-view, multi-dimensional data, and if they are single cell data, then this is a big challenge. How to integrate them and how to find out the genetic signatures. Okay, and here we also you can use some deep learning methods to use, but and also some multi-objective optimization. Previously in the feature selection method, I described the single, that is one objective optimization, but there are a lot of multi-objective optimization. You can use those optimization to find out the parent optimal gene signatures. Okay, and another thing is that the drug discovery is important things. We have to find out when we get the genes, we have to find out which drugs has a target to the genes. Okay, so and also from the cell lines data, we have to that we integrate those cell line data and drug discovery data together. This is another things that is also the molecular docking is there using for drug discovery and other things. And also the multi-objective regression model is there. Models means set of features, and we have the regression coefficient. Using this regression coefficient, we can analyze the external and also find out, we can analyze the prognosis data, patient prognosis data of survival data. That means how much chance to survive any data, and for that data we need that data. Suppose you have the cancer, if the patient is already died, for how many days he lived, and if the cancer patient are there but he already is still living, so what is the days, that means from the initially start date of the that means treatment, and the what is the current date. So all this data is there, and we can use some regression technique to analyze this data and find out the models. So this is the reference. I'm not going to the details. So this is my contact information. Anybody has a questions in the latter, if somebody want to some collaborate, they can that means see my that is website, and also my email ID. And this is some good memory. I was in Harvard. In the middle picture, that is the Harvard University campus. It's a very good things. Okay, and the right side is there. It is I went to the picnic with my team. It is in Houston. Okay, and in the right down, it is the in the California. There is a conference where I get some award. So this is a very good memory, and the research obviously is the teamwork. Okay, and that's the things. Okay, so if you have any questions, you can ask me. Okay, thank you so much.
Thank you so much, Dr. Saurav Mallik, for this extensive deliberation and making this session really an enlightening one. We really thank you from the core of our heart, from the portals of Hooghly Engineering and Technology College. For this
Thank you so much. Means if you have any
country,
question, you can ask me. I can try
uh,
my best.
I request them to write their question in the chat box. We would like to have a few questions so that we can have more engagement at the end of this session. We want an intense question answer round. So I request our dear participants to write their questions. Well, we have got a question from Mr. Dhrubojyoti Bhaumik. Dhrubojyoti Bhaumik asks, what is the main parameter or top priority for feature selection? This is
So
a
the thing is
question.
that what I said that feature selection is the basic things, right? The parameters I already described that is single objectives. Okay, if you if you know or if you want to use some multi-objective optimizations, okay, like there is a lot of NSJ is there. There is a lot of so you can still make a that is new measures or new that is feature selection method. But the top parameters are two things. Okay, one is redundancy, another is relevance. But it is not the top. If you want, you can add more things. But the basic thing is that we have to focus on the maximization of the relevance and minimization of the redundancy and make them together to make a single objective. If you want to make the multi-objective, you can add other features. But these two redundancy and relevance, these are the two major parameters. Okay.
Okay, thank you, Dr. Mallik. Any other question from the participant side? Please feel free to ask your questions. Please write it in the chat box, or you may unmute your microphone and ask questions. We are giving you access to unmute the microphone. Okay, we have got another question uh, from Shoham Das. Shoham Das, he has got another two questions. One is he has asked for your contact information. He wants to contact you later.
Yeah, yeah,
So if you could share your contact details.
yeah.
Also,
Hold on.
there is another question. Uh, Genomes cancer research on computational biology research scope.
Hold on.
Dear speaker, if you please focus on the question in the chat box.
So the thing is that genomes, genome cancer research on computational biology research scope. So the scope is higher. So I suggest if you are that means beginner in that topic, I suggest try to focus on the single cell sequencing data. Okay, single cell is a very hot topic, and there is a big challenge. And I suggest that single cell sequencing data is the future for the computational biology. Okay, and other previous things maybe it is obsolete. You can see now the big challenge is the single cell sequencing data. So this is the future of the computational biology.
Okay. Uh, any other question? Any other question from the participant side? You may either unmute your microphone or you may write it in the chat box. Well, Ratul Kumar Majumdar has asked, can we get the slides? Yes, definitely. Once we get the slides from the speakers, we will share it with you via the registered email ID. All the presentations uh, after the conclusion of this faculty development program. Well, I guess having no more questions, let us conclude this session. Uh, Thank you, dear participants, for attending this session. And one more announcement from our side that the joining link for the second session, will be shared with you at 2:45 p.m. today, and it will be shared in the WhatsApp group as well as via your registered email address. And please ensure that you have Microsoft Teams installed on your system, as the future sessions may be conducted on that platform. So make sure that you install the Microsoft Teams in your system. Also, the feedback link for both session 1 and session 2 will be shared today at the end of the session 2. Okay. We have got another question from Mr. Dhrubajyoti. Okay. Shomita Shet has greeted, like, very nice session, sir. Thank you. And she says that it is my PhD topic and I learned a lot from you. a lot from this session, sir. Okay, Shomita, hope you'll get more uh, leads from Dr. Saurav Mollik if you contact him, and stay tuned for the upcoming sessions. There will be more sessions which will give you deep insight. Next question says like, for multi-parameter based feature selection, how did you give weight for each parameter for
Okay.
multi-parameter based
The thing
feature?
is that, yeah, yeah, I'm giving the answer. The thing is that here basically there that means it actually depends what you want. Okay. So thing is that there is a lot of factors. There is a lot of factors. The thing is that sometimes if you can use any Pearson correlations value, then you can add it because in the second step that is in the redundancy collections is there. So if you want to use some Pearson correlations value, you can add that value as a weight. Okay. If the there's a lot of factors. Another thing is that if the size in sometimes in the MR case, in the MR case, they uses the size of the already selected features. So it actually depends. Okay, and here in my work, I think it is the consecutive work of the that means BWMI that is made by one of the group members. So we just following this. So if you want to that means check in which basis they are taking, I think you have to read the papers. Okay, but the thing is that don't focus on the weight. Okay, weight that means, but the thing is that in which criteria you're taking the weight, that is the main thing. Why? Research means why. You can't take randomly, that means from here. Depending upon the data, whichever you use, you have to use that means weight. Okay, that's
Hello,
my answer. Okay,
hello.
yeah,
Saurav, this is Dhruvajyoti Bhomik,
yeah,
ACMU.
huh,
Probably you remember me. Okay.
maybe,
Yeah,
maybe or may not be.
yeah. I was with ACMU. Okay.
Okay.
So, just I would
No,
like to know the how the weightage you are assigned to multiple
no,
parameter.
you have to see the paper. Maybe I forgot that because
Yeah.
it is used from the VWW MRMR, and that is made by Tapos Babu, okay, one of my friend. So I have to
Okay.
check this.
Okay.
That means the thing is that it's not my work. The thing is that uh, it is the nice things. We don't need to focus on the weight factors. But why? In which sense we have to take the weight factor? That is the things. If we use the gene expression data or DNA methylation data, we have to make some variance or whatever. It actually depends. Okay, and
Yeah.
the
Actually, the feature selections method based on the multiple parameter. No?
multiple parameters. These are the basic two things: relevance and redundancy. If you want
Yeah.
to add more, you can add. Okay, there is a no biasness or restrictions to add it. Okay,
Okay,
Yes.
yeah.
thank you, Dhruvajyoti.
Okay.
And also we have got a response from some of our participants. Like Shumit Pal says, it's a nice
Thank
session.
you.
Shubhrata Datta also mentions the same. It's a nice session. Shoham Das, Anuradha Shah, they all mention like it's a very nice session. Also informative one. Shiva Prasad Sen. Thank you everybody for showing your interest, and we again join at 2:45. Thank you for the time being, and a very special thanks to Dr. Saurav Mallik for this insightful session and making us all, uh, enlightened regarding this recent technology and its avenues, its prospects, and also the further research scopes. Thank you so much. That's all from my side, and I thank you once again from the on behalf of IQAC, IIC, and EDC of Hooghly Engineering and Technology College. This is Dr. Shubham Ganguly, Basic Science and Humanities Department, HETC, signing off. Thank you.
Thank you so much, everyone.
Thank you.
Thank you so much. Okay.
Okay, so here we leave.